Okay, picking up where we left off. Monkeying around with cancer for profit. During the early 1970s, these researchers were experimenting with various heat treatments for preparing hepatitis B vaccines from infected chimpanzees and humans. Some inactivation methods were effective, they reported, and others were not. In any case, the rush was on for immediate human trials. Following Krugman's evasive statement regarding who should be tested, Dr. Desmetier responded to concerns that a viral outbreak might occur from these experiments. He and other inner circle researchers knew that Krugman, since the mid 1960s, had been using children at the Willbrook State School for the Mentally Retarded as human guinea pigs for Army and Atomic Energy Commission funded hepatitis B studies. Jesus. In an effort to determine the lowest concentration of hepatitis B virus needed to immunize against hepatitis, Krugman inoculated dozens of New York children with what was believed to be possible live cancer-causing viruses. At the time, their rationale was that the children were likely to become exposed to hepatitis B viruses anyway. In that setting, so little more consideration was given to the bioethics of such experimentation. In relation to Dr. Prince's problem of quarantine for chimpanzees, Desmetier argued, it is certainly not unavoidable that chimpanzees become infected before or after captivity. We have been monitoring 2 colonies for about 4 years. In one, more than 60% of the animals become infected with hepatitis B virus during holding. He then told the group of another monkey colony that fared much better, to which the New York Blood Center's Dr. Prince replied, most of the animals that we have examined at the Laboratory of Experimental Medicine and Surgery in primates in New York that have been held for use in other programs for a year or more, I would say more than 70% have been environmentally infected with hepatitis B. Prince's admission that 70% of their quarantine caged monkeys had sustained hepatitis infections in some unknown way was exactly what I had feared. Being held for use in other programs for a year or more, the monkeys were undoubtedly infected with more than just hepatitis B virus. The New York group, I learned, had, in fact, been experimenting with various types of viruses and mutant strains, just as Gallo and his Littleton Bionetics associates had been doing. The only difference is Gallo focused more on type C cancer virus suspects, including ribonucleic acid retroviruses that produced leukemia, lymphomas, and sarcomas, whereas Krugman's and Purcell's group concentrated somewhat more on the herpes-type DNA viruses and type B RNA viruses associated with various forms of hepatitis and certain cancers of the liver, breast, nasopharynx, and lymph nodes. These include the yellow fever virus, cytomegalovirus, herpes simplex virus, and Epstein-Barr virus. EBV, thought to be associated with Burkitt's lymphoma, BL, Burkitt's lymphoma, most commonly seen in Africa. Their work overlapped so much that they often shared their expertise and resources, including viruses. As documented in my United States Assistant for International Development Literature Search, during the late 1960s and early 1970s, Krugman, Hillman, and Purcell held a virtual monopoly over viral hepatitis research, yet their vaccine studies were not limited to hepatitis. Their quest for lucrative grants and contracts included studying dozens of virus strains in search of highly profitable vaccines. This created a grave laboratory and outbreak threat. Their chimps became natural breeding grounds for deadly contagions, including hepatitis, herpes simplex, cytomegalovirus, Epstein-Barr virus, measles, mumps, rubella, tetanus, diphtheria, smallpox, and polioviruses. Any number of mutant viruses may have been formed as the apes lived in Guerin's and Prince's labs. This then was one plausible way in which slow-acting AIDS-like viruses developed in Gallo Bionetics labs may have accidentally transmitted to experimental hepatitis B vaccines developed in Gerens's lab. The vaccines tested in New York on humans, including homosexual males, kidney dialysis patients, the mentally retarded, and other high-risk persons during the Phase 1 and 2 studies discussed by Purcell. In 1974, a virology symposium sponsored by the Gustav Stern Foundation, Purcell and Krogman discussed their problems and progress in developing what would later become Hillman and Merck's hepatitis B vaccine. Purcell stated his failure to culture hepatitis B virus, the MS strain that Krugman had pulled from Ham, a mentally retarded child, in human cell cultures. Likewise, rhesus monkey cell cultures failed to grow the monkey-adapted hepatitis B virus. To overcome this problem, live chimpanzees were selected to grow all different types of hepatitis B virus the researchers needed for their human experiments. To avoid duplication of these experiments and wastage of seronegative, scarce, and expensive chimpanzees, we are collaborating with Dr. Barker of the Bureau of Biologics, Food and Drug Administration and Dr. Maynard of the Center of Disease Control in an interagency study of hepatitis B infections in chimpanzees, Purcell wrote. A high priority of these studies is the establishment of pools in hepatitis B virus. Human serum or plasma containing hepatitis B antigens or subtype ADW, AYW, ADR, or AYR has been inoculated into the chimpanzees. The inoculation chosen to represent subtype AYW was serum supplied by Dr. Saul Krugman from the MS2 pool of hepatitis B virus. Cross-challenge experiments and evaluation of various aspects of passive and active immunization against hepatitis B infection, Purcell explained, later proceeded in chimpanzees, rhesus monkeys, and humans. I later learned that Krugman's affiliated New York City University Medical Center was the world's leading institution for testing simian-to-man organ transplants, blood transfusions, and vaccine research. The final piece of heterogenic theory of AIDS puzzle fell into place when Purcell admitted the FDA, CDC, NIAID, NYCUMC. Purcell admitted that the Federal Department of Agriculture, the Center of Disease Control, the National Institute of Allergy and Infectious Disease, and the New York University Medical Center. What is AEC? AEC. Atomic Energy Commission. The Federal Department of Agriculture, Centers of Disease Control, National Institute of Allergies and Infectious Disease, New York City University Medical Center, and Atomic Energy Commission, Army and later Merck collaborative experimental hepatitis B vaccines destined for humans included viruses grown in chimpanzees concerning any number of monkey virus contaminants that could have given rise to HIV 2 or 1. Further implicating their hepatitis B vaccine, as Schultz explained, a lentivirus isolated from chimpanzees, SIV, simian immunovirus, is the closest primate relative of HIV-1. SIV is likely evolved then because the chimps had been, 1, used to develop experimental hepatitis B, and other vaccines largely because they were primates bearing the greatest similarity to humans, and 2, among the first creatures to be exposed to the man-made retroviruses by way of inoculating of experimental monkey cohabitation. It also occurred to me that even if Merck's human experimental hepatitis B vaccine hadn't included chimpanzee serum, only serum taken from New York's retarded children or gay men, live viral contaminants injected around 1970 could have combined with the simian virus SV40, simian immunovirus, or SFV. What was SFV? Something feline. SFV. Simian foamy virus, Simian immunovirus and simian foamy virus. The volunteers likely carried following vaccination with Merck's polio vaccine administered during the previous decade. In 1976, the Willbrook State School, under intense criticism for publicized cases of child abuse and neglect, was closed. The children, many possibly carrying the world's first AIDS viruses, dispersed back into the communities from which they came. Only the State Institute for Basic Studies in Neuroscience stayed open on the campus, explained Leonard Sayakio, a local biology teacher and historian. The neuroscience lab conducted microbiological and biochemical studies. They were studying how cells were affected by various toxins. Though now the accidental theory of AIDS seemed highly plausible, since major funding for all this work came from Merck, the U.S. Army, Centers of Disease Control, National Cancer Institute, National Institute of Allergy and Infectious Diseases, United States Assistant for International Development, and the Atomic Energy Commission. Given the Army and Merck's connections to Kissinger, biological weapon research, and COINTELPRO targeting of gays and blacks, the intentional transmission theory remained to be disproven. Though now I had identified the self-serving bureaucracy strong evidence indicated had brought AIDS into the world, I realized the hardest evidence still remained to be analyzed. The Krugman-Purcell-Hilleman Phase 1 and 2 hepatitis B vaccine lots allegedly in safekeeping at the FDA. I'm just looking up what FDA means. It might not be agriculture. Food and Drug Administration. Silly me. Agriculture. Also, look back studies of AIDS cases among Willowbrook alumni and others who received these vaccines were clearly warranted. Here's the pause it if you need longer. Yeah, that's it for today. See how beautiful fall is.